Okay, so it sounded like from the introductions that a lot of you in this room, I mean, it sounds like we have a lot of years experience here. So probably all of you have had to do, sperm searches, seen epithelial cells. So this isn't going to take very long. I'm just going to, quickly go over this. Okay, so this is something that we're used to. This is a mixture of spermatozoa and epithelia using Christmas tree staining. And you can see in the image that I have circled the sperm. This is at 20x objective. And I was so glad when after I heard Pat's talk the other day, that I had used the appropriate terminology because the whole microscope thing is something I had a long time ago and I don't always remember everything. But this is at 20x, this is Christmas tree staining, and the red doesn't look very good on this image, but everyone's seen this before. This is something I took out of this histology book. I'd never had a histology class, but anyway, this is a photo of a spermatozoon, and really of importance to us, or what we usually are concerned about, is not everything, the basal pate, the proximal centriole, mitochondrial sheath. We're more interested in the head, and very rarely the neck and tail. That thing's pretty neat. And then this is something else I took out of a histology book, Atlas of Human Histology. This is a figure of dissociated squamous epithelial cells. And generally what we see, of course, are cells that are not all together like this, but broken apart and on a field on a microscope slide. But generally, again, what we're interested in is sort of the shape of the cell, different shape, and then of course the nuclei. So here's the image we already looked at. That's the same image. So here's the 20x objective. Here's a 40x objective of the same image. So now we're starting to be able to see our sperm. And of course, you can tell as forensic scientists that this is a beautiful sample, and this is not something we see very often with the tails. So this is a fresher sample. But of course, sometimes your epithelial cells will be folded. They're not always nicely laid out like something like this. But we've all seen this. And this is the same image again, just at 63x. And here we go. Here's our beautiful sperm. Oh, sorry. And this is kind of a nice looking epithelial cell. Okay. And there's another one. Beautiful. Of course, with the Christmas tree staining, the nuclear fast red stains the nuclear material, which is what we're more interested in, and the green is just to help differentiate between the nuclear material and what's not nuclear material, and just to see the cells. Okay, this is a picture, I put just a couple other pictures on here of a mixture of spermatozoa and epithelia on a pen slide, and this is polyethylene naphthalene film, which is with the Leica microscope slides, and I think it's the same with Palm or similar, is a film that's glued by the edges onto a glass slide, and you'll see these in the lab. Okay. And what you'll notice in this image is, well, it's 40x objective. You see there's a field of epithelial cells here, and of course you see some sperm. But one thing, at least about these pen slides, is you see these pores, okay? And like they were talking about, there's an architecture to these slides, which takes a little getting used to, but it's just a learning curve. Eventually you kind of get used to looking at it that way. But there are other slides that are coming into development with like some of the feedback from Christine and some of the feedback we've given, at least to Leica, that we were interested is, was there any way we could get a slide that was nice and clear? And actually there are new slides that are in beta testing, I guess would be the correct term. Hopefully they'll work out. But for now, this is what we have to work with. And another thing you'll notice is that we have, this is a stained slide only of the nuclear fast red. And this is probably what I'm pretty sure that's what we have in the lab. It's all slides that just have the nuclear fast red. And actually I do have some slides that I brought that are Christmas tree stained. But the reason we did this was actually the studies that Christine did with staining. She did a very extensive study. And we kind of went with just the nuclear fast red staining because she did show that The green, which is an acid, very acidic, may inhibit PCR. At least, you know, she did see a decrease in peak heights with downstream analysis. So we just went with staining the nuclear material, and you know, as she suggested, I believe, with her studies. So that's why you just see the red nuclei stained. And here's a 63X objective of the same photo. Nice pictures. And so, of course, in forensic sexual assault evidence examination, we rarely have the opportunity to see samples that have tails. I mean, it's rare, I know, if I see one like that. So you can see the image down below of the sperm. It's got the head, neck, and tail. It's a beautiful sperm. And, of course, we always want to identify the nuclei of the epithelial cells. But the biggest thing is you want to make sure, if you're identifying sperm and there's no tail, you know, to me the biggest indicator is going to be the acrosomal cap that doesn't get stained. You know, because sometimes you'll see, you know, yeast on your slide, red blood cells, you know, and who knows, any number, you know, blob, a pink blob on there. And kind of like we were talking about at the previous class, sometimes you get so tired you're looking at a slide and, you know, all of a sudden you're seeing this pink blob and you're like, oh, is this a sperm? I'm tired, I can't tell, you know. So of course the acrosomal cap is key, at least, you know, in my eyes, in identifying a sperm head. So of course with laser microdissection, if we're going to actually be doing separation based on the cell morphology, we want to make sure that we're seeing that cap. So I always just do a nice fine focus back and forth to really see if I can see that cap there. And that's it. Any questions?